I think Kanika, you can admit people, and Ritu, you can look after the YouTube part and other things.
Yes, ma'am.
Yes, sure,
Just,
ma'am. Yeah.
just keep an eye on because a lot of people are admitting in short time. Okay, so we'll wait for 2 3 minutes, John. Is that okay? I mean, let people
Of
join,
course.
and then I'll introduce you because this time is, I think, people from US and largely from Europe will be missing, but I hope that people from this part of the world are joining in, and a good number is joining. Yeah. Maybe, yeah, good idea, Shwati. So those of you who can keep your videos on, can please do so. Keep your mic off, but video, it'll be nice if you can keep this on. Possible. Those who can do so, for whoever it is possible. 12 p.m. Ritu, you can hear me well?
Yes, ma'am.
Yes.
All right. So I think we should not keep Dr. John much waiting. So hello everyone from different time zones. We are very lucky to have Professor Jonathan Iredell from Australia. And this is, as those of you who are familiar with this series, this is the third talk, and this talk series is organized so that we can hear the perspective of physicians who have delivered phage therapy or developed program on phage therapy. So Dr. John is one of them. He's a doctor and he's infectious disease physician and clinical microbiologist at Westmead Hospital in Sydney, Australia. And he coordinates nationwide clinical trials related to life-threatening infections. And his research covers critical infections and microbial ecology, especially concerning transmissible AMR resistance in bacteria. And the most noteworthy part for this series is that he and his team has developed a program called Phage Australia. It's a STAMP protocol, which is the protocol required for delivering phage therapy end to end, right from isolating phages to delivering it to a patient. So we all are very eager to hear from you the how you developed this program, what were the challenges, and how has it been faring so far. So I welcome you all very much, Professor John, and for on behalf of three consortia which are organizing this, hosting this event. One is the International Bacteriophage Research Consortium, which is a joint effort by the College Acharya Narendra and Open Source Systems Limited in US. Then we have Open Source Pharma Foundation, which is co-founded by Professor John Menon, Jay Menon. And then we have the Indian Phage Society called Society for Bacteriophage Research and Therapy. And its key office bearers are Professor Vinod Kumar, who is the president, Professor Amita Gupta, who is the vice president from Delhi University, and Dr. Ramesh from um, the Vellore Institute of Technology. And very many interested audience are present here to listen to you, so I won't take much time. I welcome you once again, and thank you so much for your time and joining. Thank you.
Thank you, Amy. Thank you so much for the invitation and your kind introduction. It's quite an honour to speak to such a large audience, and an audience which obviously has a great deal of familiarity with the topic. I wasn't quite sure, Amy, whether I should how to pitch this, but it's a little bit clinical and a little bit bacteriophage, and I guess we'll just hopefully have something for everyone. So so we have seen the need, like all of you, I think, on this call, to come up with a response in terms of therapeutic applications of bacteriophages. And it's largely driven in Australia by a consortium that is led out of our largest state, which is New South Wales, and it's funded by the public sector, largely our research bodies. Like I think everyone around the world, a lot of this stuff has come out of research funding, and then groups like ours have begun to apply it a little bit more from that context. So I'm based, as you said, at a hospital, in a health department, and at the University of Sydney, where I'm a professor of medicine and microbiology. So this is probably an audience I don't need to convince about the importance of antimicrobial resistance. And of course, we're all in this region, and a lot of our neighbours have pretty severe antimicrobial resistance problems. And I don't think I need to convince this audience about the attractiveness of the natural predators. It's important, I think, to just remind ourselves of the common dogma around bacteriophage therapy, and that includes the synergies with antibiotics and their value in the presence of antimicrobial resistance, which at least in Australia has been the main driver amongst clinicians who are unfamiliar with this to seek it for their patients. I think it was helped considerably by the review, which is now, goodness me, it must be 8 years old now, this review from Jim O'Neill on AMR, which suggested the value of bacteriophages as an alternative therapy. And so it was probably about 2007 that I first treated someone here in Australia. And the status of bacteriophages at that time was that you could import it personally, for personal use. You could drink it at your kitchen table, and that was perfectly acceptable and perfectly legal. But it was difficult to have it administered in a hospital, because the duty of care then transferred to the hospital and its institution, and people were unwilling to take that on for an unregistered, regulated therapy. So we work with colleagues who were then in Amplify, now called Armata, which is based in the west coast of the US, to look at the impact of essentially a group of K-phages really targeting Staphylococcus aureus. And what we had to do was to develop a bit of a standardized approach, which is the genesis of our national program. We reasoned that physicians were used to using courses of around 1 or 2 weeks, so we completely arbitrarily set a 2-week course. We reasoned that twice daily therapeutic applications was commonplace, and so we settled on that. We reasoned that there was a desired multiplicity of infection of probably around 10 or more, if we could achieve that. And we reasoned that since septic shock or severe sepsis is associated with a bacteremia of around about 1,000 CFU per mil, or 1,000 genome equivalents per mil in sepsis and septic shock or higher in a human, then we needed to probably deliver about 10 to the 9 phages to achieve something like a multiplicity of infection of 10, or at least 1 to 10, given the dilution into a 5-litre blood volume. So that was kind of rationale that we started off with. We had a standardized sampling, which was obviously designed to collect the host response and the bacterial adaptation to the predator, and whether there was any change in the phage itself in response to being exposed to the bacterial adaptations. So that was the purpose of our strategy. And we also did some kinetics, which I think is just common sense to get an idea of distribution. So this was where we started. I think our early learnings were consistent with what was published and really probably at that time one of the most widely publicized stories in 2017 from Chip Schooley's team, in which they sort of described a clearance, which I think we're all quite familiar with, in the bloodstream over about 2 hours. And we saw this in pretty much all our patients, and you can see this kinetic curve. And as I said, we're administering 10 to the 9 PFUs over about a half an hour. And our viremia peaked at around 10,000 PFUs per mil and was cleared very quickly. One thing that's worth noting is that in those patients in whom we were running continuous veno-venous hemofiltration or hemodialysis, that the phages appeared to be picked up by the filter, unsurprisingly. We could get none coming through in eluates. And this is an example of a patient who actually received 2 courses, so went out to 28 days. But when they came off their hemofilter, as their kidneys started to recover, we started to see the emergence of virus, which had not been detectable in the bloodstream again. In this, not in this patient, but in another patient in whom we saw a similar thing, who was also on ECMO. So this is, for those who are unfamiliar with it, this is the extracorporeal membrane oxygenation technique. This is when we essentially substitute for the role of the lung by providing oxygenation to the blood through a machine. It's a different kind of filtration system, the oxygenator, and it does not have that same issue with binding to the membrane. So in hemodialysis, but not so much in ECMO, we seem to get clearance from the blood, which was much the same as we saw within, very quickly from the innate immune system in people who had normal kidney function. So I guess that meant to us that we would have to treat patients on dialysis as though they were clearing it quickly from the blood. This issue of kinetics, initially we were unsure how to deal with this, but we thought it was useful to understand it, to understand dosing. And one of the other things that we wanted to understand was what would happen if we had co-circulating virus and bacteria together in large amounts, whether we would see some resistance adaptation. So this graph, which is taken from a paper that we put out a few years ago, just shows the tracking of bacteria in the bloodstream. So this is a group of people with staphylococcal endocarditis proven on echocardiogram. These are repeatedly blood culture positive. So the solid bar means that right up to, in this case, day 3 or nearly day 4, these people, this one up to day 5, still had positive blood cultures for Staph aureus. At the same time, we measured the DNA burden of Staphylococcus aureus, and that took quite a while longer to clear. So it was evident to us that even though we were no longer able to culture viable Staph aureus in the bloodstream past the first few days of phage therapy, that we certainly had bacterial DNA detected in the bloodstream, often for quite some time after the institution of bacteriophage therapy, which went for 2 weeks and started at this point here. And the reason this is important is because we were injecting 10 to the 9 PFU twice a day when we had often thousands, you can see many thousands of bacterial genomes in the bloodstream at the same time. So if there's any setting in which you would expect to see resistance develop in vivo, you would expect this would be it. Now, this was a combination of 3 K-phages, basically, from a commercial preparation, a GMP preparation, and we really got no resistant virus detected. Although we had no resistant bacteria, although we continued to culture bacteria in the presence of the virus, these were completely normal and they were completely unchanged in their genome, and they were normally susceptible to the phages that we had infused. So our experience has been that we do not see a lot of bacterial resistance involved, which is sort of contrary to what we would have predicted, I suppose, and contrary to what we would normally expect to see in vitro. In any case, it was apparent to us that because we were not sure how to use this, we needed to establish some sort of a trial program to collect data. And this was actually quite a challenge because, and we still have a problem with acceptance in some quarters of this, because we wanted to make sure that you could accommodate what our government allows, which is essentially in the context of a great enough need, in other words, a life-threatening situation in which no other therapeutic option is available, and which our government accepts that a safely prepared bacteriophage preparation is acceptable to use, then any prescriber can use it. And so the challenge was to design a protocol that allowed extreme flexibility for any phage product over any route to be used, but data collected from it. So what we set out to do was to gather up all of the key microbiologists in the major universities and the key physicians in the major hospitals around the country and get an agreement about how we would do this as a country. And what we needed to establish was firstly some rigorous gatekeeping. That is to say, we wanted to prevent a scenario in which people started to import phage and use them in a chaotic manner. We agreed that we would start initially with the kind of protocol that I just described a second ago, which is this one, where we had a standardized course, a standardized sampling approach, and an agreed standardized dosing. Now, we could talk about dosing. Maybe that's not the best dose to start with. Maybe twice a day is not the best way to use it. Maybe 2 weeks is too long. Maybe it's too short. But we needed to agree on something so that we could standardize it. And we needed to have a gatekeeping process so that we were sure that everybody who received the experimental product, that is to say the bacteriophage, had all their other therapies optimized, but they still met the criteria of extreme need. So these were all patients like the ones I described before, who were having all their optimal therapy, but still had a high chance of dying from their underlying disease. And therefore, our government says that's fine to use an experimental therapy in that context. So that was the concept. And the protocol that I described is the one we used. We registered as a clinical trial. And the reason we did that was because we wanted to have the clinical trial framework that allowed us to monitor, to manage, and to provide, to be blunt, some sort of a sanction. Obviously, if a hospital signs on for this as a clinical trial, then the physicians involved in the trial are subject to all the normal governances and sanctions that apply to a clinical trial environment. And that's what we wanted to do. So essentially our endpoints were to understand the kinetics and the monitoring and to understand the practicalities of doing this. We set up a data safety monitoring board, just like you would for any clinical trial, which has now met a couple of times, to allow direct reporting to our national regulator. And as I said, we wanted that clinical trial framework. And of course, when we talk to people about phage therapy, like everyone, we start with happy anecdotes. And this is a lovely anecdote because it is such a multinational anecdote. This is a young lady who had a car accident when she went home to visit her grandparents and ended up in hospital with a dreadful, highly resistant infection. It was actually a NDM expressing Pseudomonas aeruginosa. So for those who were unfamiliar with it, that's just a really difficult to treat pathogen. And it was clearly involving all the metal work in particularly in her left leg. And despite removal of the metalwork, she had a persistent osteomyelitis causing a lot of pain and a lot of grief. And she got to the point where after a year in and out of hospital, the local team was seriously considering amputation, and they had discussed this with the family. How about amputation to try and get rid of this problem, get her out of hospital, get her back to school? So we reached out internationally and were met with a response from Ran Nirpaz and Ronan Hazan from the Hebrew University of Jerusalem and Hadassah Medical School, who furnished a terrific bacteriophage, an anti-pseudomonal bacteriophage, which they arranged to be purified from some colleagues in New York who are at APT, the commercial operation there. And they provided that to us, and we treated this young lady. She had 2 weeks of therapy, and from the very first consultation and request, right through the period of the international outreach, the response from Ran and Ronan, the purification in the US, and the installation of therapy was 6 weeks from start to end, and she had a wonderfully successful outcome. So we often present that as one of the great anecdotes about international collaboration, and I don't think we can emphasize that highly enough. So she did really well after a couple of weeks of phage therapy. This was a couple of years ago now. She's off all her antibiotics, and she's feeling well and back at school. We learned a few things in the process, much as we had intended to do. This is on the left-hand side for the physicians in the room who are familiar with looking at this stuff. This is the classic before and after MRI. I think these are T2 images of a resolving infection. She had quite a problematic osteomyelitis that was clearly not going anywhere before her phage therapy intervention. And this graph on the right is a trajectory of time right out to beyond a couple of months. And on the vertical axis, you can see the bacterial genomes per mil. So we were doing this by qPCR, and you can see that soon after she started bacteriophage therapy, these are these downgoing grey arrows here. That's phage dosing. Soon after she started that, she had a spike in her C-reactive protein. So this is a sort of an interleukin-6 surrogate for those who are unfamiliar. So it's a classic inflammatory marker in the bloodstream. And with that, she had little temperature. She had a bit of local pain in her leg, which she hadn't really experienced a lot of in the week or so prior to coming into hospital for therapy. And soon after that, she had a spike, a flush of Pseudomonas DNA detected up to nearly 2,500 genome equivalents per mil in the bloodstream, which slowly subsided until we increased the dose again, whereupon she had another flush of pseudomonal DNA into the bloodstream, less of a fever spike this time, less of a symptomatic response, and this just slowly subsided. And we get that classic predator-prey curve that follows. So the blue curve here with the arrow attached to it is the number of genome equivalents of PASA 16. So this is the phage we got from Ran and Ronan and gave to this young lady. So we had the classic predator-prey response, and interesting that flush of bacterial DNA into the bloodstream in a little girl who'd never been bacteremic and who had no such DNA in her blood at baseline, and which disappeared subsequently. Her inflammatory markers after that initial spike slowly settled. So we got the impression from the end of the bed that, we'd done something with the phages here. And that this is that sort of auto-dosing phenomenon, which we talk so much about in phage therapy. So when we looked at her inflammatory transcriptome, so we did RNA sequencing on this young lady's blood in the same, remember I showed you that time frame we used to collect samples, and we asked the question, was there any inflammatory response? This is too messy to go through, but suffice to say there are pro-inflammatory and anti-inflammatory response genes that waxed and waned. This is the pre-phage period, and you can see that the pre-phage period is closest to the period some 2 weeks later, but is quite different to the immediate early treatment where we saw that big inflammatory response. And if you look at these markers, we don't have time to go through it today, but these markers were the classic markers of the innate immune response, the markers of the TLR4 pathway, which we would expect to be highlighted in the presence of LPS and those sort of pyrogens, and matched perfectly consistently with the little fever spike, the CRP spike, and that flush of bacterial DNA. So it kind of confirmed our sort of anecdotal suspicion that we had lysed a whole bunch of Pseudomonas in the bone, flushed it loose into the bloodstream, caused an inflammatory response, which was detected as a CRP, a temperature, a bit of pain, and a classic transcriptome response. And soon after, if you go to the bottom part of this, you can see this adaptive response. Remember I pointed to 2 spikes, and of course we had, the innate response, and of course we had an adaptive immune response, because remember, we're injecting 1,000 million viruses twice a day directly into the bloodstream. So we might expect an adaptive immune response to that. And I'll come back to that in a minute. So early on, we also asked our local prescribers what they thought of all this, because bacteriophage therapy is not well known in Australia. And even now, we are really the only site in Australia that is doing this. We have some colleagues who are just starting, who've treated 1 or 2 patients in Melbourne, and some colleagues in Perth who are starting shortly. But the majority is just being done at one site. And most Australian physicians are quite unfamiliar with it and therefore skeptical. And they nevertheless had heard about it. They thought that it was probably safe if it was properly prepared, and they wanted some sort of a protocol that provided timely access and rigorous data around these key aspects of therapy. And when we asked them when we asked them simultaneously what were the key targets that were most important, then clearly these were the ones that we all might suspect. Those things that are resistant, including the dreadful Mycobacterium abscessus, and of course, those things that are just difficult to treat even when they're not very resistant. One of the arguments we had to make to government was about cost effectiveness. So in Australia, we can deduce what the government thinks is cost effective by looking at those things that the Australian government has subsidised in the past and asking the question, what did that cost? And how many quality adjusted life years benefit do you get for your dollar spent for that intervention now that it's subsidised? And the answer is, it's around about 35 to 40,000 Australian dollars per quality adjusted life year. So that seems to be the kind of thing that is around the ballpark of what the Australian government thinks is reasonable value for money. Even though they don't set out with this at the outset, it's surprisingly consistent when you go through all the different interventions. And it's actually much the same number, interestingly, as you can calculate, I believe, in the UK. So we get similar sorts of numbers from the UK. So I think at least this is a common story. And if you look at common things we do, like coronary artery bypass grafting, you can see it's a little bit on the expensive side, which I think as physicians intuitively knew. If you look at intensive care services, we think they're good value. Bone marrow services are great value. And kidney transplantation is good value. These are the things that we think are must-haves in a fully developed health system. And if you look at the consensus, published consensus of efficacy for phage therapy, it is probably something along this line for a quality adjusted life year. So that in terms of a public health investment, we would argue that this is good value for money. It's as good value as bone marrow transplantation. It's a lot cheaper to do, and it's probably a must-have in the health system. If you look at it the other way and ask the question, well, let's take a single example. And in this example, we chose infected hip and knee replacements. And these figures will vary enormously from jurisdiction to jurisdiction, depending on, the sort of nature of your health systems, depending on what it costs to do these operations, depending on how many you do in a country. But clearly, there are big savings to be made in our country if phage therapy was only 60% effective, which was kind of the published efficacy we got for salvage therapy. In one single year, we would have made these savings. And that was a big deal in a small country like Australia. And our population is 25 million. So we're not a huge population at all. So those are big savings. And while we're on this idea of making the cost effectiveness argument, the other thing which we always have to bear in mind, and something we always point out to government when we're discussing this, is that this is something where we need to ideally achieve economies of scale. So while we often have trouble with making this argument, in that on one hand we talk about the value of precision therapy, and we say things like, this is the ultimate precision antimicrobial. You can just pick off a single subtype of a single species and leave everything else intact. Isn't that terrific? But in the same breath, we're then forced to admit that, yes, well, we may often be using a whole batch only on one person. And because this is batch fermentation, we can make 1,000 doses or 10,000 doses almost for the same cost of a single course. It's just a bioreactor after all. And so it's essential to achieve economies of scale. But if you have a system in which you run a surveillance program and you know in advance which are the subtypes of your various high-risk species, so your golden staph, your Pseudomonas aeruginosa, your E. coli, your Klebsiella, your big streptococci. These are the classic killers in sepsis. If you know in advance what subtypes of those species you have in your country or in your region, then you can predict in advance the kind of phages you will be asked for if you were to use phages to treat them. And if you can therefore match your production system with a surveillance program, you only need to use about 10% of the batch to get down to the sweet spot. Even 5% of the batch really dramatically reduces the cost per usage. Because the system is simply one of matching the right phages to the right bacteria. And so I think this matching program, or this biobanking program, is something that we believe is really important, and we've spent a lot of time and energy on developing, because we argue that this part is actually not that complicated. It's certainly not as difficult as making, a CAR T cell system or a gene therapy system. It is actually, with the right combination of chemists and microbiologists, you can actually do this pretty efficiently, I believe. But this part is the tricky part, because you have to reach these economies of scale if you can. So everybody on this call is probably familiar with the difficulties of choosing the right phages. But for those of you who are not, if there are any less phage savvy physicians on the call, just to remind you that we typically define them or historically define them in terms of their relative efficiency against some sort of a reference strain. So if we have a nice reference strain and we've got a nice phage and we can reliably count on X number of spots per plate when we get the phages and the bacteria to meet in predefined concentrations, then we compare that relative activity from bacteria to bacteria of that particular phage as the efficiency of plating. And that's the sort of time-honoured approach. The other time-honoured approach is, of course, growth inhibition in liquid culture. And this, for many of us, feels more familiar if you're someone who's more familiar with antibiotics and antimicrobial drugs and MICs, then this is the kind of data that you're more used to looking at. And we often look at the sub-MIC concentration so that we can detect the antibiotic synergy that phage provides. And this kind of information is the sort of thing that we routinely provide. Now, this is actually borrowed again from one of Ranen's publications, but just to point out that all the time we are dealing with forcing difficult trade-offs. And so if you make it easy for the bacteria to squirm away from the phage attack by simply changing a receptor that may be easy to change and doesn't hurt it much, then it will easily adapt. If you then attack it with a second phage that maybe uses a different receptor or maybe attacks the alternative receptor that might be offered, then the trade-off, the fitness cost becomes higher. And if you then add in an antibiotic, which requires a totally different adaptation, you're creating a fitness landscape which requires more and more difficult trade-offs. And that's essentially what we're trying to achieve. So we're always aiming to get to this point where it's just really too hard for the bacteria to adapt. And we present this as synergy testing to the pharmacy. So at the moment, the way we've set it up locally is probably like most people do, I think. We try and make it look like a traditional doctor-patient interaction with some sort of a diagnostic test, notwithstanding the caveats that we're all familiar with and which I just alluded to briefly. We then provide something that looks like an antimicrobial resistance test result. And then we have, at least in our country, the requirement for meeting the compassionate use guidelines. So it must be someone in whom this therapy is justified. We seek informed consent to use it because it's an unregulated product. And our country uses a system that is something like the European system or the Belgian system, at least, of the magistral phage concept, in which you have essentially a compounding pharmacist or a manufacturing prescriber, which is an extension of the concept of preparing an ointment in your surgery. A prescriber is allowed to take responsibility in many countries, including ours, for a preparation which is formulated by them for the treatment of their patient. And you can use this kind of wrinkle in the regulatory system to provide phage therapy or a phage prescription. We then have to satisfy, of course, the pharmacy or the drug committee of the relevant jurisdiction, in our case, say a hospital, for example, who would then dispense this material that we've provided, that it is safe and appropriate. So we normally produce a characterization of the phage itself, and everybody around the world, I think, is familiar with having to provide a characterization that the phage is not inherently lysogenic on the basis of either the phenotype or maybe quicker if it's cost effective, and it is for us, the genotype. Similarly, the presence of antimicrobial resistance genes, toxins, and other undesirable elements of the phage need to be ruled out to the satisfaction of the drug committee. Likewise, the strain in which it is produced, the manufacturing strain, clearly there is a risk that co-purified prophages will emerge. This is actually a really common problem. If they're present, you have to address them, characterize them, describe them, and assure the drug committee that the risk associated with them is not important. And similarly, you don't want to be co-purifying toxins and AMO and goodness knows what else if you can avoid it. This is a bit of a difficult topic and maybe one that we can discuss later, but suffice to say that's our approach, and I think a common approach. And of course, there's a batch, the manufacturing safety production of the batch, which we normally do endpoint sequencing on. And again, the depth to which you sequence is important if you want to deal with co-purified prophages in your mixture and other impurities. And so we do endpoint sequencing to reasonably high depth, typically at least 1,000x of the target phage. And we need to assure the regulator that it's 99.9% pure on the basis of genomic purity, at least in the preparation. Our sort of network at the moment, as I said, is largely national, and we've agreed to approach this as a national consortium for the sake of uniformity and consistency, and that's really important. And also really important, I think I made this point before, I talked about RAN and RANN. I mentioned the European approach. Jean-Paul Pernet is well known to everyone, I think. And Rob, we've got phages from them and a lot of advice, like many people from the Belgians, about how best to approach this, and indeed from our colleagues in Poland and Georgia about how to approach this more generally. So it's very important that we treat this as an international collaboration. That's why it's so lovely to have this opportunity to speak to you all about how we do things. And I hope you don't recoil in horror and tell me that's a crazy way to do things. But at least in our country, this is our local consortium, our national consortium. And it's worth calling out the global phage case runs that we do, which is very similar to this, I think, which RAN is involved with, and Greg German and Gina Saar. The biobanking network I alluded to before, I think it's important, and Amine Katami, who did the phage stuff. Now, I've seen, Umi, you've come back on video. Is that because you want me to wind up? Because I have a few more slides, or I could stop here if you prefer.
No. I was like writing notes, so I was trying
to
be
okay.
distracted.
I thought you were going to give me the big wind up.
No.
Okay.
No. Please go ahead.
Okay. I'll just go a little longer because I wanted to leave time for discussion. But this is just to give you an update of where we are in our little country. And it's really, this is just our site. I mentioned that our colleagues in Melbourne who've got a terrific integration of basic phage biology and clinical medicine there, and our colleagues in Perth who are really a respiratory medicine unit who are very, very focused on treating lung infections. But most of the stuff in Australia is still coming out of here. We've been sending phages to multiple states in Australia, and we have a lot of hospitals now in the STAMP program that we provide phages for, much as I think many centres do in other countries. So our referral rates are sort of generally rising. We don't advertise. We try to keep it quiet because we actually are already right at our capacity to handle the referrals. But fortunately, we're going to increase our capacity quite substantially in 2024. And I think we clearly need to. There's no doubt that there's increase in this all around the world. The referrals that we get, I suspect, are similar to everyone else's. We get a lot of bone and joint referrals. A lot of them are Staph aureus, but probably the majority are still the really antibiotic resistant ones. And so I think there is a boom in prosthetic joint infection that we're yet to be hit with, but we've certainly treated quite a few Pseudomonas and Staph bone and joint infections. In fact, the little girl that we gave an example of was one of those. Chest infections are a common one. It's either Pseudomonas or it's Mycobacterium abscessus, and of course we're very grateful to Graham Hatfall and the team at Pittsburgh, who very generously provide therapeutic phages for abscesses, many of which are genetically modified. So we've sent kids home here in Australia with genetically modified phages to be delivered at home by nebulization on many occasions. So it's increasingly well received. And then there's a sort of a distribution like you'd expect, I think, for the other sources of infection. In our the latest version of our study, which has got much more rigorous data collection, because you remember I said to you before that we treated 14 patients in the staff study. 13 of those we published, and one more that we didn't include in the publication. It's a bit late. And we've treated another 14 to date on the stamp, and there was probably about half a dozen in the transition. So we're now heading towards patient number 40, but not quite there yet. So of the 14 treated in the latest iteration of stamp, clinical response, I don't want to make much out of this because there's no control group. There's not even a case control group defined yet. So although I've got this in bold, it's a bit naughty of me because I really shouldn't. It's all just anecdotal data. What we're interested in this study really is collecting the kinetics and the adverse events. And we've really had in the Staph aureus study, although they were all critically ill and on life support, we couldn't see any adverse events. And then in this group, who are not critically ill, we haven't seen any adverse events either. And that's, I think, just because we've got nice clean phages. And now, we make all these ourselves now for local usage. The bacteria we treat, I think we pointed out before, the sites, and as I said before, still dominated by SUD because it's often quite resistant. A lot of chest infections, particularly CF kids, CF community, cystic fibrosis community, at least in Australia, is classic early adopter community. So dosing and duration, I said before that we made a guess about this, and it was based on published animal data. So we thought it felt sensible. I didn't know what number to target, honestly, but I knew that our studies of bacteremias in septic shock during the ARISE study, which is an investigation of early goal-directed response to septic shock that we did in Australia. When I looked at those data and the genomes per mil of bacteria and that's how I sort of made this deduction. And I pointed out that it's cleared within hours. And in fact, a lot of people use this kind of dose. It's relatively easy to produce it in a mill, and it feels like a sensible dose. So it's probably a typical kind of dose, I think, in a lot of people's hands, certainly in ours. I mentioned that often the phage has a bit of a tail, but certainly the viable phage is undetectable within only a few hours. We typically, initially we tried to readjust dosing, which was probably a bit foolish actually, but we did initially try and dose adjust on the basis of the amount of phage genomes present before the 24-hour mark. And we did that because we wanted to understand, it's very expensive to do all this, monitoring and qPCR and genomics, you can imagine, but we had to make the investment to understand what we were doing. And the bottom line is that the twice daily dosing appears to be about right, because we really, in most people, the phages are just disappearing, at least their genomes are just disappearing from the blood. We don't detect viable phages much after about day 3 or day 4. And we do see an adaptive immune response emerge and peak not long after, and that may be relevant to that. Certainly all the animal models, I think it's reticuloendothelial clearance that we expect to be doing most of this. I'm not going to talk about adaptive immune responses and the sort of really interesting question of whether there's some immunomodulation. We published a tiny bit about this, but I think these publications are better. If you're interested in that topic, I think it's a really exciting area. But we have looked in some detail in our protocol about the antibody development. And so I mentioned before that adaptive immune response that we saw in the transcriptome from the little girl that we treated with the Pseudomonas osteomyelitis. We see that sort of response repeatedly in our patients now, but that was just a nice clean example of it. And we see neutralizing antibody develop fairly quickly. This is a reflection of the reduction in the Phage EOP, which is a reasonable surrogate over time. And you can see that there's a few logs of drop. By the time we get to about day 7, certainly by the end of 2 weeks, we have got an effective EOP when you add serum to it that implies neutralizing antibody development. And if you look at neutralizing antibody development in that first study, that Staph aureus study, then we can see really quite a consistent response. This patient had a bone marrow transplant and really was not making an immune response to anything much. So we do see quite consistently matching the transcriptomes that I pointed out to you before, this classic adaptive immune response. And this is something that has been published for the abscessus experience as well. So this is Beck, Dedrick, and Graham Hatfull published a while ago. I should point out, however, that I'm being a little bit glib here. I don't want to tell you that this is entirely predictable, because it appears to us that, in fact, some patients are not mounting an adaptive immune response at the end of a week or so and are not developing neutralizing antibodies at this level. So we're not sure, and maybe some of the audience can tell us, but I'm personally not sure whether this is the individual, whether this is a characteristic of those particular phages, or whether it's just a bit of a wrinkle of that particular infection. Certainly those people who can't make an immune response, you can see, but, and in the Staph aureus, these very sick people, there was a bit of a pattern, but some people just do not seem to sustain a good neutralizing antibody response. But it is probably true that effective therapy may be limited, at least in some people, by neutralizing antibodies. We think this amplification effect is a useful guide in vivo. So a qPCR that shows you this big boost of phages that occurs in our hands, typically around day 4, day 5, I think it tells you that you're hitting the target. So that's useful. And I think a lot of us think that, you know, testing, some way of testing how painful it is for the bacteria to wriggle away and adapt to the phage. If you can do that in your phage susceptibility testing in advance, I think that's probably useful. I mentioned we use this sort of compassionate use protocol. Our mission is to inform the regulators here so that they feel comfortable about developing a regulatory framework. And I said to you that the cost effectiveness of this whole story is super insensitive to economies of scale. But if we want to preserve the precision antimicrobial story, then we have to link it with a surveillance program. Otherwise, we're going to have to take the one-size-fits-all approach, which, to be fair, I think most of our commercial colleagues have said, look, this is just common sense. If I'm going to make a phage therapy against the golden staph, I'm going to get a bunch of phages, and I'm going to make sure that the bunch of phages that I put in my single ampoule will cover most of the golden staphs you're likely to meet. So I think that is sort of a halfway mark, maybe between true precision therapy and a targeted surveillance program. What we're trying to do at the moment is make sure that if we do combine phages together, that we have the choice of the prescriber being able to choose them on the basis of susceptibility testing and make a synergistic cocktail. That needs us to routinely report the resistance development and the receptor, the target for the phage, which is what you hear Paul Turner and John Koff and Ben Chan argue in favour of for Pseudomonas, where they'll say, look, it's only just common sense. If you're going to give a 3-phage cocktail against Pseudomonas aeruginosa, you ought to send something after the the pilus receptor, something after the LPS receptor, and something after something else, so that it becomes very difficult. That fitness landscape is very difficult to navigate. So I think that we probably need to have that routinely in our surveillance program. So our biobanking scheme includes this is nascent, I have to say, it's not complete yet, but we are including the receptor site for each phage so that we can choose them sensibly when we start making cocktails. And that again is just, I think, common sense for us. Look, I'll stop there. I think it's 25 past. And I'll thank you for your time and ask if there's any questions.
Thank you, Professor John. That was excellent. Very well explained. And as you can see in the chat, there are several questions for you. But I'll take the privilege of being the moderator. So first question to shoot will be me, and then I'll take everybody else's questions. So is that okay? And I hope you have time to take all these questions. My question is more general. I had so many, but given the questions in the chat, I will skip them. But mine is, like you said that you have developed the stamp protocol, and so to take everybody on board, whoever was involved, whether from the government, regulatory authorities, and the other collaborators, how could you sail through all of it to come to a compassionate program acceptable to all?
Stormy waters, I think, to continue this sailing analogy. But I think, in a small country like ours, we can go to the infectious diseases and microbiology community. So that includes the clinical people as well as the leading scientists. And I think it's essential that both communities are part of this, obviously. And it was just a little bit of persuasion, but not much, because I think everybody pretty quickly agreed that it was just a common sense approach. And so it wasn't actually hard to convince everyone.
That's nice. And in case of that young lady, you could do the therapy with one phage only, to pass the 16. You didn't have to try a cocktail or more than one phage, right? For her entire course?
Well, that's true. And I have to say that because you sometimes can't get more than one hit, you hope that the phage you're given is one that is difficult for the bacteria to handle. So the phage that Renan and Ran had provided was really just too much for this particular bacteria. Now, that may be a bacterial burden issue, because remember, we're using antibiotics as well. And it may be that this young lady just needed something to tip the scale, and the fitness landscape for that Pseudomonas was already too difficult. But clearly, I think we'd all agree that we would want to make it as difficult for the bacteria as possible. So we want a complementary attack between phages and antibiotics whenever we can. Unfortunately, I have often had to use single phages in combination with antibiotics. But if we can demonstrate really a straight line in the growth kinetics with the phage and the antibiotic that we're using, I feel a little bit more comfortable that it's difficult for the bacteria to adapt.
Completely. All right, so I'll take questions. This is Dr. Taruna Anand, and her question is, what is the best route of giving phages in case of bacteria infecting and residing in lymph nodes? How do you reach lymph nodes?
Lymph nodes, that's a good question. Look, we've given it by almost every route. We've given almost all of our patients intravenous, and I guess that comes from perhaps a simplistic assumption that intravenous delivery is the logical way to send an antibiotic in that, the bloodstream eventually perfuses all the relevant sites. I would suppose that if I was trying to treat lymph node infections, I would probably use an intravenous route, but I have to say I have no data for that.
Okay, Dr. Sriram wants to ask, will it be possible to share the stamp protocol for the purpose of awareness? I think you published?
Yes, absolutely. We'll provide all of our documents to anybody who wants them. They are available online on the Phage Australia website, and they've been published in BMJ Open, so you can just download them that way as well. They're pretty available. But yes, of course, we'd be happy to provide any template documents that would be useful.
Thank you. Dr. Adamu Kaki Kabo, he wants to ask for collaboration. So I think Dr. Kaki Kabo, if you're here, then you can ask your question. We can unmute you. He wants to basically ask, is there any channel for collaborations or support to new entrants into phage research?
Well, I think most scientists and physicians would welcome it, and I'm no exception. I certainly welcome collaboration, and we've had enormous support from our colleagues in Europe and in Israel and in the US, and the UK benefited enormously from it. I think it's essential that we have international collaboration. The only problem that we have had, to be fair, is that as phage therapy is sort of becoming more discussed, many of the university lawyers have been increasingly anxious about the potential value of the phages to a university. And some of the attempts that we have had to collaborate have been bedeviled by very constraining material transfer agreements. And that has been difficult. I think we had some wonderful experiences early on, which I gave you one example of. But I think it's important for the scientists and the physicians to find a way to explain to their organizations that they will benefit rather than be harmed by collaboration. Now, I think that's a bit of a challenge at the moment, actually.
Dr. Neha Singh wants to ask, Dr. Shahid Jabil wants to know the freezer stability of phage products. I mean, how stable they are, their thermal stability. Because to him, it appears that that could be an important factor for commercial viability of phage therapy.
Yes, that's a really good point. So the short answer is that it's a little variable. It's not clear to us exactly what the most vital variables are. It is clear that it may be a component of some phages, and it is clearly a component of storage conditions. But we can tell you that purified phages in high titer, held in our refrigerator at 4 degrees, most of the ones that we've used, most of the tailed, classic sort of Cordoveralis type phages that we've used are stable for a year or two, no problem. So we've got, we're in the middle of a testing program and we're hitting the 2-year mark, and we've got really good stability. I think that the difficult transition is that transition from storage to final product. We often find that our final product in our current dispensing in borosilicate actually goes off pretty quickly, and we're not sure whether we may have chosen the wrong type of glass or the wrong material. Maybe silicon glass is just a bad idea to store our phages in. But certainly they're very stable in a plastic Falcon tube for 2 years at 4 degrees at 10 to the 10 PFU per mil. We're just trying to work out what the variables are. But I think that means that if you can make a good batch, you can have it for 2 years. We can take it from that point into the patient's arm in 5 days. So we should be able to shorten that, because most of that 5 days is our 5-day sterility test. Once we have a program that we've demonstrated is clean, then we'll have a diminished requirement, like a GMP program, for example. We'll be able to potentially get into patient arm within a couple of days or less.
Dr. Neha also has similar question. I think Dr. Neha, your answer is here already regarding the stability of phage preparation and regarding the titer of the phages to be administered. Is it like you said 10 to 9? So is that constant for you or it's variable?
That's what we use. We, as I said, chose that slightly arbitrarily, but the kinetic measurements we've done, I think to some extent have vindicated that choice. There are those who would argue, look, the best trick is to just give a whole lot of phages all at once and stand back, just, light the fire, so to speak. And there are others who would say, well, just give a little bit and give it for a while. I think we use more, for example, than the Queen Astrid protocol. We use less than others in the US. And I can't really defend the dose other than to say we all needed to agree in Australia on standardized dosing so that we could measure what we were doing and have it reproducible. Maybe when we analyze all our data, in a year's time, when we've done the first 100 patients, we'll say, you know what, we should have given 10 to the 7, or we should be giving 10 to the 10. And we shouldn't give it for 2 weeks, we should give it for 5 days, or we should just give 1 dose. I don't know because we haven't done that analysis yet.
But you did this excellent CRP analysis, the course tracking CRP levels. So perhaps if you change the doses and see the CRP, you might know that which causes least inflammatory response, but still is effective.
Yes, that's right. We may well do. The trouble is our patients are so heterogeneous. I think we'd probably need a cohort of patients with the same infections. And so I suspect the really good quality data is going to come from clinical trials. I know that Armata is currently running a dose ranging study with Staph aureus. So they will probably generate some pretty good quality homogeneous data in a cohort of patients.
Thank you. So another question on dosing. So Malvika, I think that got answered. Our next question is more technical, that when you want to study the synergistic response of phages with antibiotics, so do you give the mix of the two as a single preparation, or both are given separately at their own regular intervals?
That's a really interesting question. The co-formulation question is a difficult one. We do have a colleague who is ligating antibiotics to the phage capsid head, so that if we use that approach, then we can do microdosing. And we're just starting to do in vivo trials with that. And that is probably a classic co-formulation scenario. And we are also using fairly standardized approaches for intracellular phage therapy. So in human phagocytes, for example, if you were to treat Shigella, then we would co-administer, and maybe they should be co-formulated. But I guess I would take a leaf out of the Belgian playbook and say that at the moment, it would be reasonable to co-administer at the bedside, but always to be able to choose your own preparation of phages. And I think that's different to what we're seeing emerge from the US jurisdiction, where we're seeing co-formulated phages. It may be that from some of those companies we'll see co-formulated phages plus antibiotics. There's clearly going to be a market to rejuvenate some of the orphan drugs or the drugs that have gone off patent through different formulations. We may see that sort of thing emerging. Yeah, it's a good question, and I don't have a good answer.
It'll come soon. This is from Smriti, and her question is specific to MRSA, methicillin-resistant Staph aureus. So have you given, she's asking which countries are legally following phage therapy against MRSA?
So which countries are using Staph aureus phage therapy, you mean? Was that the question?
Yes.
Yeah.
Especially the methicillin-resistant Staph aureus.
Yes. So I know that in the US, the Staph aureus is a big target for 2 of the big commercial companies there. I'm not really very familiar with their operations, but I do know that the people that we originally worked with, now Armata, working hard to get a staph cocktail through the FDA, and they are currently running a 1b/2a trial, which means presumably their next trial will be a phase 3. There is talk of introducing a Staph aureus arm, or we've applied to introduce a Staph aureus arm, a Phage arm to the Staph aureus network adaptive trial, the SNAP study, which is running, I think, in Australia, Canada, Singapore, Israel, not sure where else, but so there are some multi-center network adaptive platform studies. And I think the network adaptive platform trials are probably the best vehicle to start to get some signal from phage therapy as adjunctive therapy. And they certainly do include the multi-resistant Staph aureus.
Dr. Kopala, do you want to ask questions directly?
Yeah. Thank you very much. Dr. Jonathan, my question is that the genetically engineered phages, what will be the stability when they are used in vivo?
So the genetically engineered phages that we have used have been exclusively those engineered at the University of Pittsburgh, and mostly they have been things where lysogeny genes have been knocked out, really. And they have been perfectly stable in vitro as far as we can tell. A lot of it's about the formulation. So some of the formulations are coming lyophilized, and it's not clear to us how much of a benefit you get from freeze drying or spray drying. Some of the phages come in glycerol at really cold. We had, for example, the PASA 16 that Ran and Ranen provided via APT came in glycerol at -80. So I presume that reflected concerns about stability. Now, that was a non-modified phage, and as far as I can tell, the genetic modifications that most people are doing should not be affecting stability.
Okay.
Shiva Mishra wants to ask, what was the reason that only 16% patients were cured while 83% have shown clinical response? This was to your side.
Yeah, so we only defined a cure if someone had complete microbiological eradication and complete clinical recovery. So most of our patients are referred in extremis with a lot of underlying pathology. And so if someone's still in hospital, even though we might have achieved microbiological eradication and they've still got multi-organ failure, they were not deemed a cure. I think that reflects the cohort that we were receiving. In our Staph aureus study, we had a predicted mortality on the basis of the available scoring systems, which are not great. You know, we used the Apache score, we used the infective endocarditis scoring system, and we had a pretest mortality of 40%, but an actual mortality of 28%. I'm not going to pretend that that was significant, but I think that while you're working with a heterogeneous cohort in which you're only doing salvage therapy, then the expectation of a cure is very low. Most of our patients are referred, you know, after months and months of failed surgery and antibiotics. It's very hard to fix anyone. I think it's the same sort of scenario as when the first organ transplants were done, when the first heart transplants or liver transplants were done, and people arrived in catastrophic end organ failure. And they're the sort of infections that we're treating at the moment. I'm hoping that our regulator will say, actually, we think this is safe. We'll allow you to do less sick people. And then I think we will start to see better quality signals.
Right. So another question, follow-up question on stability. When we deliver, so typically the phages, as you mentioned, are given either in saline or glycerol stock. So do you recommend the carriers for phages like liposomes or some kind of encapsulation that might make them more stable? This is from
That's a really good question. At the moment, I guess we feel that because it is so easy to make a preparation, and because our current shelf life at 4 degrees appears to be approaching 2 years now, we haven't felt the need to improve stability. But yes, that's a really good point. For phages that are intrinsically unstable, lipid encapsulation, nanoparticle encapsulation are sensible things to think about. Absolutely.
So would you recall that famous case of phage burnt patients where I think that clinical trial went kaput because the viability of phages had got down. There was nothing wrong in the protocol, but perhaps they forgot to check the titer or viability, and that's how the whole thing went bad. So
The Phage study, yes,
Phage burn
yes, yes, that was a terrible story, wasn't it? But yes, that does raise the question of your cold chain logistics. So we have only really tested this once, but we made a preparation of phages and sent it around Australia to ask 2 questions. Firstly, can we get it there safely? And secondly, how fast can we get it there? So we were able to get it there within 24 hours to Perth, which is the most distant site, 4,000 kilometres away. And in every site that we sent it to, we essentially had retained viability. Now, that's not the same as saying we had stability a month later, but we do know that in our own stock, as I said, it's lasting very well. So we test our final version of phages in the glass ampoules. We test them every 28 days, and a certain percentage of them have lost a log or 2 in titer at that point when they're prepared at 10 to the 9 in SMB in borosilicate.
Sure. Dr. Gunaraja from Nepal, his question is, he again wants to ask, he wants to also know this, that if there are any possibilities of collaboration with Phage Australia on phage therapy trials or tech transfer. He's especially interested in how can we extend this to low-income countries like Nepal. He wants to be part of the...
No, we'd be delighted to collaborate with scientists, physicians anywhere in the world. And I think I made the point before how fortunate we were to have had the links we've had to date. And yes, we would absolutely love to collaborate with anyone here. You've obviously got a wonderful network yourself here. And we tried to do much the same thing as you've obviously done so successfully in our global Phage case rounds. So yes, we would be delighted to collaborate with anyone and everyone. The sort of things that I think would be really nice to do together would be to look at really simple multi-center trials. I think you know there are companies now that are trying to do this already, and we need to be conscious of not duplicating effort. So one of the good things to do, and again, you have a terrific forum to do this, is to understand from your perspective, all of you on this call, what is the most important study that you want to see done, and how does it differ from the sort of studies that commercial companies are offering right now. So, you know, the things that come to mind in our own country, for example, are some of the regional problems that we see, the multi-resistant gram-negatives, you know, the Acinetobacters, the Burkholdeриas, those kinds of things, which are so problematic all around the world, but particularly in the more temperate and tropical climates.
Yeah, so Dr. Kunaraj, I think you got your answer. He's very active phage researcher from Nepal. Swati Shom is a science communicator, and her question is, are healthy humans exposed to bacteriophages in the bloodstream? She's if the patients are being exposed to phages in their system in the first time. So she wants to know whether when you administer, is it their first time exposure or healthy humans are already exposed to bacteriophages?
Yeah, well, that's a good question. I mean, all the Phage researchers on the call would be, you know, used to rolling out the the story about how, the phages are the most numerous and diverse life form on the planet, and that we've all got more phages in this than we've got our own cells, and probably at least one phage per bacteria, and that we know that bacteria routinely spill into the bloodstream. It's normal human biology. And I don't think any of the phage biologists in the room would think that there aren't phages circulating through the human bloodstream routinely. I guess the thing that we do when we give phage therapy is we give these really high doses. I mean, I don't think we ever expect to find a phage viremia in your average person walking down the street at 10 to the 5 phage genomes per mil of blood, which is what you get after you get an injection of them. So we just on that point, because that's a really interesting question, we half expected to find an anamnestic response to phages, some of the common phages, things like Staph aureus, for example, where everybody meets a Staph aureus every now and then, and the phage diversity in Staph aureus is actually relatively more limited. And so therefore we might predict that, hey, most humans have probably met an anti-staphylococcal phage once or twice in their life. We couldn't see an anamnestic response, but maybe when our data are more numerous and better quality, as our numbers get up, we'll start to see a signal there. But I think for things like SUDS and the other things, probably the capsid diversity and the tail fiber diversity is enough that the antibody response is probably not going to be commonly anamnestic. I don't know, actually. That's a real interesting question. We don't know the answer to that yet, I don't think. Ask me again in a year. I might have some data then.
Yeah, sure. Thank you. Iredell wants to know if he finds in some literature that Pseudomonas isolates form, which are susceptible to antibiotics, but this forms strong biofilms. What could be the reason for Pseudomonas isolates forming biofilms in spite of being susceptible to antibiotics?
So that was a question about SUD. I mean, they're great biofilm formers, aren't they? They're one of those classic sort of non-fermenters that have these, type 4 pili that we know are classically associated with initiating biofilms. And I think they're not all there is, obviously, to biofilm formation, but they are a bit of a question when you're applying phage therapy, in that we know that some features of some biofilms, I mean, the most famous example, I suppose, is the curly of E. coli or some E. coli's, can deliberately confound a phage attack in a biofilm. But as a general rule, I often tell students and doctors who are less familiar with clinical microbiology that those organisms that are well adapted to living in a stressful environment, like the non-fermenters, so we're talking about Acinetobacter, Pseudomonas, all those sort of things that we think hang out in drains and vases by the bedside, they're really good at biofilms because they're adapted to live that way. Whereas those things that are really well adapted to a competitive environment, like a mammalian gut, and like everything to be 37 degrees and have some sugar and have some food to eat, like an E. coli, for example, they're maybe not quite so good at that sort of thing. It's probably true that most bacteria, when they're pushed, develop something that looks like a biofilm. We're never quite sure what to call things, are we? We call them persisters, and we call them, stringent forms, and we call them biofilm states, and we call them L forms, and probably everything we look at is a bit of a mixture of all of the above. And they are difficult to deal with because the testing we do for phages is always done in the logarithmic growth stage. That's what we extrapolate from the antimicrobials, where we always do logarithmic growth testing. And the curse of that, of course, is we know that some antibiotics, the MIC for an aminoglycoside for a Pseudomonas in a biofilm is at least 1,000 times higher than in a vegetative growth stage. We don't know what that number is for phages, and it may vary from phage to phage, actually. but the nearest surrogate we've probably got is things like L forms, which grow reasonably well, although they're not really easy to adapt to microteter plates. But again, that's data that will come in the next year or so. So it sounds like a weak response, but ask me again in a year.
So we are like right now fixing a year after a talk with you. So much you can see the chat. I mean, so many thank you messages. Really appreciating your talk. I mean, including of course myself. Dr. Ankush, would you like to ask your question? I'm not able to figure out. We can unmute you. It's regarding some page typing. Ritu, can you unmute please, Dr. Ankush?
Yes, ma'am. Yes, ma'am. He can unmute himself.
Yeah, please go ahead.
Hi, Professor Jonathan, for such an enlightening talk, and it was very thought-provoking. Thank you for that. I have a question that you said that you are working on screening of phages and bacterial receptor host receptors. So this is the most difficult part, that when a patient samples come, the patient is already in a very bad shape, and the time is lost while screening and then preparing the phage preparations which are actually working on the patient. So can't we do the phage typing with respect to the specificity determinants like their tail fibers, etc., which are binding to the specific receptors, because you have very few categories of receptors on which the phages tail fibers are binding. So can't we do that typing and we prepare an in vitro assay so that we can quickly screen for those phages which we have in our library, and we can straight away go with this phage cocktail and then do the treatment?
Yes, absolutely. I think that's a really good point. There's no doubt that if we had to do a huge, extensive characterization every time we chose a phage, we'd just not be in a clinically relevant time frame. And it is true that there is an opportunity in the diagnostic space. You can imagine sort of phage display concepts, fixed membranes, cloned receptors, you can imagine all sorts of diagnostic opportunities. A lot of people are exploring those, including us, because we're all wanting to save time. But I guess the low-hanging fruit in my own mind is that, coming back to our screening program, if we know the subtypes of Pseudomonas or Staph aureus or E. coli that we're most likely to face, and we've got a bank of bacteriophages, we can define their receptors in advance. And we can say, well, look, we had to choose phage 12b. That really likes, a type 4 pill. And so that's a good one to have in this mix. And then there's that other phage over there that really likes an LPS receptor. So let's put them together. So that's kind of what I meant. And I think that's what the team in Yale are arguing for their classic cocktail for their Pseudomonas, Yeah. So I think your point's very valid. Yeah.
Yeah. So this will be an advanced form of phage therapy that will be precision medicine for the patients. I hope. Yeah. So hopefully we have some time, 2-3 more years to develop that. So thank you for that.
Thank you, Dr. Ankush. So Shoma, your question is also, I think, answered here regarding customized preparation versus the cocktail. Now, Dr. Geeta has a question that, have you had any experience vis-a-vis treating an eye infection with phage preparation?
Topical, you mean?
Topical, yeah.
Yes, we haven't actually treated a topical infection, but yes, I mean, goodness, that must be a classical application, mustn't it? Pseudomonas in the eye. I guess one of the questions we always have to ask ourselves is, have we got an application in which there is an already effective response? So I guess the eye surgeons would say, well, look, Pseudomonas, dendritic ulcers, we can use topical aminoglycosides, we can use topical quinolones. Sometimes we have to do surgery. Would the phages be better? That's a good question. Do we think that more rapid killing or more effective killing will make a clinical outcome? That's a good question as well. Do we think that the phages will work better in biofilms and be a good way to salvage an infected lens or other site. The truth is, you probably just throw it out, wouldn't you? So always we have to ask ourselves, is there a true need for it? I think at the moment, pursued mostly the biofilm scenario, people are crying out for it in lung infections, particularly cystic fibrosis, chronic bronchiectasis, chronic bronchitis, and in bone and joint, I guess, is the other classic biofilm sort of scenario. But yes, you're right. The eye infection is another classic one.
All right, I think still questions are coming. You have to tell me, can we take more or should we close it and maybe they reach out to you directly? I think, it tells your talk has evoked such response. Endless question. I thought this was the last and then I find this yet another question.
Well, we've got a few more minutes. It's up to you,
Okay,
Rami.
wonderful. No. I have all the time for you. It's just that if you have, sure. Then
Sure.
this question was from Nutan Shukla. She has a question. Have you ever experienced combination therapy to improve phage therapy? I think this question is taken. Combination here, I think she means synergy with antibiotics.
Yeah, synergy with antibiotic and phages are always desirable. There is a little bit of a debate, I think, about how best to use it, but I think most people who work with phages and indeed most bacteriologists would probably agree that we should be making it as hard as possible for the bacteria to adapt. And that means putting as much pressure on that fitness landscape as possible. So, if you have a phage that is completely suppressive in logarithmic growth in a growth media, and as we were just trying to match someone today, for example, and we had 4 or 5 phages, all of which gave complete suppression in growth, and the scientists are asking, well, which ones should we put together? And it strikes me that the answer is always going to be the ones that would complement each other. But if you can't demonstrate that in vitro, you can only infer it from their receptors and their mechanisms of resistance. So I would always advocate for combination therapy just because I just don't want the bacteria to have an option.
Right. Attack from all ends, right? This Arnav has a special question. It's an interesting one. He says that at times they don't find any lytic activity against bacteria by phage, but it is still fine to inhibit biofilm formation in terms of drop in OD. So how does that happen? That phage not showing lytic activity on plate, but
Yeah.
on blank.
That's a really good question. So we were just talking about this the other day when we were doing some matching, when we got conflicting results with different growth media, and I was, all the bacteriologists and all the phage biologists on the call would be perfectly familiar with the notion that in different growth circumstances, the bacteria are probably expressing different surface proteins. If your phage has got a specific receptor that is only available in certain circumstances, then it will behave differentially according to the percentage of the population that expresses it. So you might express, let's say, for example, you target the OMPK36 outer membrane porin in Klebsiella, which is a common phage receptor, and you put that organism in a growth medium which is optimal. There's plenty of food to eat, and so the large channel porin is closed, the little OMPK36 is expressed, the phage eats the bacteria because it recognizes that receptor. You then take that same organism and put it in a much more stringent growth conditions where there is much less nutrient availability. OMPK36 is no longer expressed, OMPK35 is expressed, maybe the phage doesn't recognize OMPK35 because it's OMPK36 specific, and you appear to have lost all your phage effect. So I think it's completely unsurprising that we would see this in different growth conditions. The challenge for us as microbiologists is to figure out how best to test the susceptibility and predict which growth conditions are relevant in vivo.
I think you got his answer. This was, I think, the last question of the session, the technical question. And then I think, Swati, you can ask your question directly, because through you, I think you can reach out to many who are perhaps not as well versed with the jargon. But your question on the customized preparation and the cocktail preparation, I think, Dr. John, if you can answer, it would be great. This is from Ubaidu. Before that, one question. Ubaidu Rahman wants to know, is there any effect of antibiotics on the stability of phages? Like when you give them together. I don't think they're stored together, but when they're given together.
Yeah, that's a good question too. I mean, some of the antibiotic preps contain things like chelating agents. And if we rely on things like a bit of magnesium in our buffers to improve stability of capsids, and then we add in EDTA, which comes with the antibiotic that then picks up the stabilizing divalent cation, we might affect phage stability. I don't think that's been well tested, and maybe that's one of the reasons that I'm a bit conscious of not co-formulating things that might potentially do that. So yes, I would expect it would be relevant. It's probably, I guess off the top of my head, I would think the excipients are the ones I'd worry about more than the chemicals themselves. We do, as I said before, do link certain antibiotics to phage capsids using a specific ligand, and that has proven to be very stable and not compromise them. But we've only done it with quinolones, using a nanoparticle, and with aminoglycosides and polymyxins, all of which have a nice available amide groups which can be linked easily. So yes, it could be a problem. I suspect it's specific. We haven't had any trouble with it, and I would not routinely co-formulate them unless I was sure that that was an issue.
All right. So Swati wants to ask your general question vis-a-vis customized preparation, or are you answered?
Hi, I was just wondering if you expect that each patient will have to have their own specialized, customized phage therapy, or will there be a super effective phage or a phage concoction that can be given to multiple people? Whoever has Pseudomonas or whoever has staph infection can be given the same concoction.
Yeah, good question. Certainly the staph is a case in point. So I think that the team at Armata are betting that their concoction is going to be a bit one size fits all. I think they have 90+% coverage of the staph strains they've tested. Every region is going to have possibly a little bit of variation, given that the diversity in the staph phages is a bit less, and Staph aureus is maybe a little less variable on its outer membrane than, say, you know, a coliform, for example. Then it might work for staph, but it may not work for something like Acinetobacter. So I think it varies with the bacterial target. In our own experience, which has been dominated, as you saw, by more than half of the requests being for Pseudomonas, in our experience, we have got one or two phages which we've used several times. And so we do have some favourite phages that seem to be used repeatedly. And of course, once we've got experience with them, we tend to keep serving the same one up to the drug committee because it makes them more confident. You know, oh, you've used that before and that wasn't a problem in patient 2, so you can have it for patient 3. So there's a little bit of psychology going on there as well. But the biological question, I would expect that some types of receptors are going to be less variable than others. So if you choose, say, an OMP36 receptor in the example I gave before for Klebsiella, then that would apply very widely, I think, across Klebsiellas, because OMP36 surface domains are not that variable.
So are you thinking of developing a bank within your hospital also, like that you have clinical isolates and having phages against all of those clinical isolates, so that they become handy for you, in time of emergency?
Yes, we do. By lucky circumstance, we happen to be the reference I also look after the antibiotic resistance reference service for our state, and I do the I support the genomic surveillance for the gram-negative bacteria for our country, the Australian Surveillance Program. So it means we have a really good understanding of the bacteria that are turning up in the bloodstream in our state and in our nation, and a good understanding of, because they're all sequenced, so a good understanding of their genomics, and we can test them. Our national surveillance program now, for example, wants us to do phage matching against all of these, so that we will soon understand, and this will be a job for next year, but so that we will soon understand what the best sort of phages or what the common phages requests are going to be.
I think that will be really wonderful. It doesn't require absolute new infrastructure. I think the existing infrastructure just needs to be supported a little.
Yep.
All right. So with that, we come to end to this very wonderful session. We can't thank you enough. I mean, it was awesome. I mean, you your collection to treat patients outside. Yeah, one last question. Geeta wants to know, have you tested your phages against Australia anytime?
Outside Australia, we haven't had any requests to send phages outside Australia at the moment. We certainly have sent them around Australia a little bit now. I think we're probably, excuse me, not as well known as some of the other services, such as the Taylor Lab, which does provide phages all over the world, and the Queen Astrid Medical Hospital, military hospital, which produces phages for a lot of people. So I think international export is not particularly difficult as long as you've got your cold chain sorted out.
All right. With that, we close. Thank you so much. Thank you very much on behalf of all of us. I thank all the participants here for excellent questions. I mean, they made the session go very nice and very well answered all the questions, for your patience, for very clearly explaining. Thank you so much. And I thank Ritu, who has been organizing the backend team, and Kanika also. So thank you all so much. And we'll have our next session next week from Indian doctors. You have Dr. Gopala, who is here. So we plan to close this by end of this December, end of this month. But it was, I think, very engaging and very informative. So I think the purpose is quite very well served, I believe. So given the response, excellent response, especially at this time of the day, you know, usually by evening we can get more people, but we had a very fantastic attendance also. Thank you so much, Ron, so much for your time and explaining it so well. I'm sure everybody will echo to what I'm saying.
Yes, thank you very much. I'd love to stay in touch.
Thank you. Yeah, sure. All right. And hope to see you sometimes in India. Thank you. Bye-bye. Thank you, everyone.